All living organisms respond to external stimuli. When bright light falls on your eyes, don't you blink your eyes? If you get a tap on your knee, your lower leg suddenly stretches forward. You start shivering in chilling weather. All these examples show that our body responds to external stimuli. Even plants respond to external stimuli. A plant bending toward a light source called phototropism, a climber growing along a support called thigmotropism, roots growing towards the direction of gravity called gravitropism are all examples of response to stimuli in plants. To bring about these responses, different body parts coordinate and work with each other. If this coordination was absent, survival of organisms would have been difficult. Let's compare coordination in plants and in animals. In multicellular animals, coordination is achieved by two methods: chemical coordination and nervous coordination. However, plants lack a nervous system. Only chemical coordination exists in plants. This chemical coordination is brought about by hormones. Hormones synthesized by plants are called phytohormones. They control the growth and development of plants.
Plant hormones belong to 5 major classes: auxins, cytokinins, gibberellins, abscisic acid, ethylene. Let's learn more about each one of them.
We are aware of phototropism in plants. The stem bends towards the light source. But have you ever wondered how the stem bends itself? When light falls on one side of the plant, the phytohormone auxin gets accumulated on the darker side of the stem. Auxin causes the cells in the dark region to elongate more compared to the cells on the other side. And this is why the stem bends towards the light source. Auxins are growth hormones. Indole-3 acetic acid, IAA, is an example of naturally occurring auxin in plants. Dichlorophenoxyacetic acid, 2,4-D, is an example of synthetic auxin. The most significant role of auxins is cell elongation. Auxins also promote apical dominance. Apical dominance is the influence of the apical bud in suppressing the growth of lateral buds. Can you guess how gardeners maintain the bushy appearance of plants? They cut off the apical or terminal buds. Auxin concentration is very high in the apical bud region. When the apical bud is cut off, auxins that are concentrated in this region are also removed. Hence, the lateral buds are no longer inhibited. So, the plant attains a bushy appearance. Abscission is the process by which a plant drops its fruit, flower, or leaf. The process is usually associated with a decline in auxin levels within the organ to be detached. Auxins, when present in low concentration, stimulate root initiation on stem cuttings.
Let's move on to cytokinin. Cytokinins were first discovered in the milky endosperm of coconut. Cytokinins are a group of growth substances whose primary effect is to stimulate cell division. However, these effects are not seen in the absence of auxins. Cytokinins together with auxins regulate root and shoot development. In tissue culture experiments, a high level of cytokinin in relation to auxin promotes shoot formation, whereas a high level of auxin in relation to cytokinin promotes root formation. As the name itself implies, cytokinins promote cell division or cytokinesis in the presence of auxin. Cytokinins help the plant to overcome apical dominance and promote lateral bud formation. cytokinins delay senescence. They also promote chloroplast development. Zeatin is an example of naturally occurring cytokinin. Kinetin is a synthetic cytokinin. Highest concentration of cytokinins are found in plant roots, young leaves, developing fruits and seeds, that is, the meristematic regions of the plant. A Japanese plant pathologist, Kurosawa, was working on fungal infected rice plants, which grew abnormally tall. It was later discovered that this abnormal increase in height was due to a substance called gibberellin, produced by the fungus Gibberella fujikuroi. Since gibberellins are acidic compounds, they are also called gibberellic acids, Ga.
Look at these two cabbage plants. Cabbage plants are supposed to be short and bushy, aren't they? Then, why is this cabbage plant so tall?
This cabbage plant was treated with gibberellins. Gibberellins cause stem elongation in intact plants by promoting internodal elongation. Dormant seeds don't germinate even if the environmental conditions are favorable. Gibberellins break seed dormancy and induce germination. When gibberellins are applied on flowers, it leads to the development of seedless fruits. Development of seedless fruits is called parthenocopy. Some plants, called long day plants, require long photoperiods for flowering. When gibberellins are provided, the flowers bloom even when the photoperiod is decreased.
Let's now learn about abscisic acid. It's interesting to note that abscisic acid, ABA, got its name because of its property to induce leaf and fruit abscission. ABA is a growth inhibitor. It induces seed and bud dormancy. Abscisic acid also delays cell division and cell elongation. It inhibits shoot growth. It stimulates the closure of stomata and thus reduces the rate of transpiration. Mangoes ripen in the summer season, but many a times you might have observed that mangoes are available in the market even before their season. How is this possible? Mangoes are artificially ripened by spraying them with ethylene. The most important role of ethylene is in fruit ripening. Ethylene also helps in breaking seed and bud dormancy in some species of plants. It speeds up leaf senescence. Ethylene inhibits stem elongation. It promotes flower and fruit abscission. Did you know that unlike other plant hormones, ethylene is a gaseous hormone? Can you guess why fruits ripen faster if they are wrapped in polythene bags?